Welcome to this lecture on fatty acid oxidation or beta oxidation. This is an important topic in lipid metabolism. The objective of this lecture will be to explore its definition, identifying locations and conditions under which it operates, identifying enzymes and coenzymes involved, the mechanism, products, regulation, and significance of this important metabolic pathway. To understand beta oxidation, it is important to identify beta carbon in a fatty acid such as palmitic acid, which has 16 carbon atoms. The carbon atoms in this molecule are numbered starting from carboxylic group designated as 1, next will be 2, then 3, 4, and so on and so forth. Another way of nomenclature of these carbon atoms is using Greek alphabets such as alpha, beta, and gamma. Alpha carbon atom is carbon atom number 2 and beta is carbon atom number 3. So basically the beta carbon atom is the third carbon from the carboxylic group in a fatty acid. The shorthand form of fatty acid is written as R, representing the hydrocarbon chain followed by the carboxylic group or the functional group COOH. However, in this lecture I will use shorthand form as R, CH2, 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 COOH, having the first 4 carbon atom in this molecule. And the reason for that is because beta oxidation mostly occurs at the carbon atom number 2 and 3. Now we know beta carbon atom, therefore let us define beta oxidation. This is called beta oxidation because it occurs as beta carbon atom in a fatty acid which is corresponding to carbon atom number 3. It completes in 4 recurring reactions catalyzed by 4 distinct enzymes and produces one acetyl-CoA, which is a 2 carbon atom unit, one FADH₂ and one NADH. Because of the production of FADH₂ and NADH, fatty acids produce more energy than the carbohydrate and proteins or amino acids. The remainder fatty acyl-CoA is shorter by 2 carbon atom than the original one. At the end of each round of beta oxidation, the remaining fatty acyl-CoA repeats these reactions until it is completely oxidized. Let us identify the products of beta oxidation and their fate. These are 3 molecules which are produced during fatty acid oxidation or beta oxidation, namely FADH₂, NADH, and acetyl-CoA. Out of these 3, acetyl-CoA undergoes Krebs cycles, producing 1 molecule of FADH₂, 3 molecule of NADH, and 1 molecule of GTP. FADH₂ and NADH drive electron transport chain to produce a large amount of ATP. Let us explore the subcellular location and tissue type where beta oxidation occurs. Beta oxidation occurs in mitochondrial matrix in the tissue such as adipose tissue, skeletal muscle, heart, and liver. Heart is the only organ which uses fatty acid as a major source of energy. Skeletal muscle also utilizes fatty acid at rest and also during extensive exercise. However, in the tissues such as RBCs which lack mitochondria or brain which does not allow transport of fatty acid into brain through blood-brain barrier, this metabolic pathway is absent. What are the major sources of fatty acid for beta oxidation? These are 3 sources. First is fatty acids coming from dietary triglycerides. Second major source is fatty acids coming from adipose tissue triglycerides. And the third one is intracellular triglyceride droplets in the skeletal muscle. Free fatty acids released from adipose tissue bind with albumin and are delivered to the oxidative tissue for beta oxidation. When does beta oxidation occur or under what condition beta oxidation operates? Glucose is the primary energy source. However, when these glucose stores are depleted, fatty acids are oxidized by beta oxidation and provide energy, and that happens under the conditions such as extensive exercise, fasting, and starvation. In addition, heart is the only organ which uses fatty acid as the major source of energy fuel. In addition, skeletal muscle also utilizes fatty acid during rest and during extensive exercise as a source of energy. Let me explain the mechanism of beta oxidation. Before beta oxidation, a fatty acid undergoes two important reactions. These reactions will be explained here with the help of this cartoon. As you know, the location of beta oxidation is mitochondrial matrix. However, fatty acids are present in the cytoplasm. They are in inactive state. Therefore, the first step is to activate them in the form of fatty acyl-CoA. So, fatty acyl-CoA is an active form of a fatty acid. The second step is transport of these fatty acyl-CoA into the matrix, and that is through a pathway known as Carnitine shuttle pathway. Once the fatty acyl-CoA is delivered into mitochondrial matrix, it will undergo beta oxidation. So activation and transport of the fatty acyl-CoA to matrix are essential and prerequisite for beta oxidation. Fatty acids are activated in the cytosol by an enzyme known as fatty acyl-CoA synthetase, which utilizes coenzyme A and equivalent to 2 ATP molecules as source of energy, producing fatty acyl-CoA, which is an activated fatty acid, and this fatty acyl-CoA is transported via carnitine shuttle into the mitochondrial matrix for beta oxidation. Carnitine shuttle system is a transport system which transport long-chain fatty acyl-CoA from cytosolic compartment into the mitochondrial matrix so that it can be utilized in beta oxidation. This system is consisted of three enzymes which are associated with mitochondrial membranes, outer membrane and inner mitochondrial membrane. It consisted of three distinct enzymes. The first one is CPT1, which is associated with the outer membrane, and it converts acyl-CoA into acyl-carnitine by releasing coenzyme A. Acyl-carnitine is subsequently translocated across the inner mitochondrial membrane utilizing the second enzyme known as CACT or carnitine acyl-carnitine translocase. This is an enzyme associated with inner mitochondrial membrane. Acyl-carnitine in the matrix, however, cannot be utilized. Therefore, it must be converted back to acyl-CoA utilizing the third enzyme known as CPT2 or carnitine palmitoyltransferase 2. This enzyme utilizes coenzyme A and regenerate acyl-CoA by releasing carnitine. Carnitine is subsequently translocated into the intermembrane space and cytosolic compartment. So the carnitine shuttle system is made up of three distinct enzymes, CPT1, CACT, and CPT2. Once fatty acyl-CoA is delivered to the mitochondrial matrix, it undergoes four successive reactions. The first one is oxidation, catalyzed by acyl-CoA dehydrogenase. Second reaction is hydration, catalyzed by enzyme known as anoyl-CoA hydratase. The third reaction is also oxidation, which is catalyzed by beta-hydroxyacyl-CoA dehydrogenase. And the last reaction is thiolysis, which releases a 2-carbon units as acetyl-CoA by an enzyme known as thiolase. I will discuss these four reactions in detail in the next slide under the mechanism of beta oxidation. I will discuss now mechanism of beta oxidation, which occurs in mitochondrial matrix and completes in four successive reactions. The first reaction involves the formation of a trans fatty acid which is catalyzed by acyl-CoA dehydrogenase. This is the first reaction, and this is also oxidation reaction. It utilizes coenzyme FAD, which is reduced to FADH₂ by extracting hydride ion from alpha carbon and beta carbon atoms. So this forms a trans fatty acyl-CoA, which is a high energy intermediate. This is not a trans fatty acid, it is a trans fatty acyl-CoA which is not released into the environment. This is known as trans delta 2 enoyl CoA. Delta 2 here means the double bond is start at carbon atom number 2 in this molecule. So this is trans fatty acyl CoA. It is not trans fatty acids. Trans fatty acids are not produced in humans, plants and animal. Although the intermediate as trans fatty Acetyl-CoA is produced in beta oxidation. This is an example of that. Now in this next reaction, there is addition of water molecule which is catalyzed by an enoyl CoA hydratase forming beta-hydroxyacyl-CoA. So this is kind of oxidation also. I have shown beta carbon atom in this cartoon with green color just to make the things clear. So this is hydroxylated fatty Acetyl-CoA. This undergoes further oxidation utilizing enzyme known beta-hydroxyacyl-CoA dehydrogenase. This also utilizes NAD coenzyme which is converted or reduced to NADH. This extract hydride ion from beta-hydroxy forming beta-keto-acyl-CoA as the product. In this molecule, the oxidation is occurring at beta carbon atom. That is why it is called as beta oxidation. There is another important point to note here. The bond between carbon atom 2 and 3, or you can say alpha carbon and beta carbon, is labile, and this is cleaved by thiolase, which uses coenzyme A, releasing a 2-carbon unit known as acetyl-CoA, and the remainder of the fatty Acetyl-CoA, which is shorter by 2 carbon atom. This fatty Acetyl-CoA repeats these reactions until it is completely oxidized. So these are the 4 reactions which describe the mechanism of beta oxidation. In summary, beta oxidation: fatty acids are activated by acyl-CoA synthetase to fatty Acetyl-CoA utilizing equivalent to 2 ATP molecules per reaction. Fatty Acetyl-CoA then enters the matrix via Carnitine shuttle pathway. The beta oxidation thereby starts or completes utilizing 4 recurring reactions catalyzed by 4 enzymes in the matrix. Each cycle of beta oxidation produces one acetyl-CoA, a 2-carbon unit, one NADH and one FADH₂, and a shorter fatty acyl-CoA. Due to production of NADH and FADH₂, fatty acids produce more energy than the carbohydrate or protein. The shorter fatty acyl-CoA undergoes a repeated cycle until it is completely oxidized. To calculate the amount of energy or ATP produced from a given fatty acid, it is important to know how many cycles will be required to completely oxidize a fatty acid such as palmitic acid, which has 16 carbon atom. You have seen that in each round or each cycle of beta oxidation, a molecule of 2 carbon unit as acetyl-CoA and 1 molecule each of NADH and FADH₂ are produced. Therefore, in this molecule of palmitic acid, you will have 7 such successive cycles to completely oxidize this molecule, producing at each cycle 1 acetyl-CoA and 1 molecule each of NADH and FADH₂. At the end of 7th cycle, there is 2 carbon unit which is also released as acetyl-CoA. However, important point to note here is that NADH and FADH₂ at this step will not be produced. Therefore, this palmitic acid will require 7 cycles, 8 acetyl-CoA and 7 molecules each of NADH and FADH₂. Using the following formula, you can quickly calculate number of cycles, number of each products including acetyl-CoA, NADH and FADH₂ for a given fatty acid such as palmitic acid on complete beta oxidation. For example, number of oxidation cycle or beta oxidation rounds will be equal to number of carbon atom in a given fatty acid divided by 2 minus 1, which comes out to be 7 cycles for palmitic acid. You can also calculate number of acetyl-CoA by dividing the number of carbon atom in a given fatty acid by 2, which comes out to be 8 molecules for palmitic acid, and the number of FADH₂ and NADH can be calculated by multiplying number of cycles by 1, which comes out to be 7 molecules each. So using these formulas, you can quickly calculate number of the cycles, number of acetyl-CoA, FADH₂ and NADH2, and you can calculate the total number of ATP produced from a given fatty acid by Krebs cycle and by oxidative phosphorylation or through electron transport chain. Let me test your knowledge. Here is a question. In how many cycles will the stearic acid and 18 carbon fatty acid be completely oxidized in the mitochondrial matrix? Choices are 7 cycles, 8 cycles, 9 cycles and 10 cycles. You can use this formula. Number of cycles is equal to 18 divided by 2 minus 1. The correct answer therefore will be B, 8 cycles. Thank you very much for watching this video. Please like it, share it and subscribe it. Until next time, you take care. Bye bye.